Standard deviation is the natural scale of a patient population. It tells you how much patients typically deviate from the population mean. The spread is expressed in the same unit as the clinical measurement. Standard deviation inherits its structure from variance across the patient population. Taking the square root restores the result to the original clinical unit. The mathematical structure underneath is unchanged. Only the unit is restored. Shifting a clinical reference range does not change the spread of the population. Converting unit scales S by the same conversion factor. S is scale equivariant, sensitive to the unit, not to the reference point. When two independent patient cohorts are pooled, their variances add. The standard deviations of the cohorts do not simply add. The pooled spread is the square root of the combined variance, not the sum of the two spreads. Two patient populations can have the same standard deviation and completely different clinical profiles. S does not capture skewness, bimodality, or the distribution of extreme responders. Standard deviation requires the second moment of the distribution to be finite. For populations with extreme responders, the second moment may not stabilize. When S exists, it characterizes the bulk of the population, not the clinical extremes.